So Ravi mentioned he'll talk over, but I actually added a few slides. So I'll start talking and then Ravi can add or take over when he recognizes his slides again. So a little bit about retroviral life cycle. So essentially, this is just a nice simple diagram showing how lentiviruses enter the cell and replicate. So essentially with HIV, your infection first begins when the viral envelope protein makes an interaction with a cell receptor and actually enters the cell. Because it's single-stranded RNA virus, the RNA genome gets reverse transcribed into viral double-stranded DNA, which then burrows its way into the nucleus, and then it becomes integrated into the host genome using integrase. After there's been accumulation of that synthesized viral proteins and genomic RNA, viral RNA, all those components then differently packaged and then they bud from the cell and then mature when GAG and the GAG polyproteins are cleaved by protease. So the problem with HIV is that it's quite highly diverse. There's a high replication rate and could be 10 million to 100 million different virions per day. When the single-stranded viral RNA is transcribed into double-stranded DNA, that process is actually, it has no proofreading abilities, so different mutations can be introduced into the double-stranded DNA. And then HIV is also quite highly recombinant. So, I mean, we've seen that looking through our PANGEA sequences. We have, you know, lots of different circulating recombination viruses. So because of those three different things, there's a large accumulation of mutations or those mistakes. Sometimes they can result in sort of nonsense mutations. They don't do anything, missense mutations. Other times they can actually lead to drug resistance mutations. So even in the absence of drug therapies, you can get some drug resistance. So that's why the modern day therapies, they recommend a triple therapy. So, I mean, all of you know this. Here's just a quick overview of the HIV genome, the different proteins and whatnot, genes. So GAG, POL, ONV, everyone knows this. There's lots of different approved drug classes. These each work on a slightly different part of the replication cycle. So first of all, we have the entry inhibitors and the fusion inhibitors. They work by stopping the viral particles from binding to the receptors. There's the reverse transcriptase inhibitors, which works on reverse transcription, two different classes. There's the non-nucleoside reverse transcriptase inhibitors and the nucleoside reverse transcriptase inhibitors. Then you've got the integrase inhibitors, which stop integration from happening. And then you have the protease inhibitors, which stop viral protein synthesis. And then you also have another class called boosting agents. So they don't actually have any specific effect on viral replication. They just increase the activity of the other classes of drugs so that you don't have to take such a huge dose. So there's been a couple of different changing HIV regimens over time. This is not exactly to scale, so the dates are just sort of there when these first happened. So a long time back when HIV was first discovered, they finally had a target for treatment. There was this monotherapy, which was zidovudine. Unfortunately, because it was just a single drug, resistance would accumulate quite rapidly as a result of drug pressures and all of those different mutation and diversification things I just spoke about. So fast forward to 1997, that's when dual therapies were first introduced. And that was two different NNRTI drugs. So commonly it was lamivudine and zidovudine. Fast forward again, there was the switch to the triple therapies. And then nowadays it's changed from, instead of two NRTIs and an NRTI, it's changed to two NRTIs and an integrase inhibitor. And then in South Africa recently, they've adopted a slight variation, and they've also had some new third-line regimens approved for a national rollout in the near future. So there's this idea of the selective pressure of therapy. This is where Ravi's slides come back in, so he can start talking over me if I miss something. So essentially how drugs and drug resistance work is, if you're taking your drug, you'll eventually start to decrease viral load by killing off those HIV virions which are susceptible to that specific drug. But then you'll always have a small proportion that are already drug resistant. unfortunately, what tends to happen is you start to get an increase again in viral load because you've killed off all of those drug susceptible viruses, but then it's actually the drug resistant viruses which then begin to proliferate and increase over time. And this is when your regimens start to become ineffective. And that's the reason for swapping to second line regimens.
That's a very nice introduction. Thank you, Steve. So this is some data from some years ago now when I first got interested in drug resistance. And this was a comparison of a number of different studies. On the top is where no monitoring was undertaken. In other words, you just started your ART and then you essentially just carried on taking it and you went to the clinic and maybe had a CD4 count occasionally, but you just carried on. And then on the bottom is places where you have usual monitoring of viral load, in other words, 3 to 6 monthly, and you can see the prevalence of drug resistance at 12 months after initiation. You can see there's much higher prevalence of drug resistance in the places that were not very well monitored. And this, we think, is explained by the figure on the right, which is a demonstration that, of course, the longer that you are, your virus load is detectable, the more you are able to select for the mutant viruses that are resistant. And this makes perfect sense, especially for drugs where you just need a single change to confer high-level resistance, such as the non-nucleoside drugs, which were used up until very recently.
So this is kind of talking about what I spoke about before and the sort of pharmacokinetics of drug resistance. So if you split this into 3 different steps, first of all, when you have no detectable drugs, then you have majority wild type. Then when you start to have the resistance occurring, you start to have the more resistant quasi-species which begin to take over, and that's usually as a result of suboptimal drug levels, not sticking to the, not having good adherence to the regimens and such. And then once you start to change and you get something that works for you, this is what we have, therapeutic drug levels, that's when the virus starts to become suppressed. If you continue to take drugs and your adherence is generally good, then that means that the virus remains suppressed and resistance is not likely to emerge.
Okay, so here is a meta-analysis we did, not a meta-analysis, actually a retrospective cohort study of a number of different patients, about 2,000 spread across a number of continents. Encircled are the data from sub-Saharan Africa, showing that actually if you do a cross-sectional survey in patients on standard ART who have not been monitored, you find in those failing, so around 15 to 20% of patients had viral loads that were above 1,000 copies. And when you sequence those individuals of virologic failure, you can see at least half of them have triple class resistance or triple drug resistance. In other words, resistance to all 3 drugs in the regimen, not just one. And this is particularly notable for the drug tenofovir, which is a drug people once believed that you could not become resistant to this drug based on clinical trials. And here you can clearly see that is not true if you give the virus enough time to evolve against pressure. Go to the next slide. So it's important to distinguish what I've just described, which is acquired drug resistance on therapy. There's also a distinction to be made between transmitted drug resistance and pre-treatment drug resistance. So on the right-hand side here, you can see a pharmacokinetic graph showing you that if you take 1 pill of efavirenz, tenofovir, and emtricitabine, that's or a triplet or a single tablet regimen, as it's called, and you just take the single pill, 2 of the drugs will fall away very quickly, but efavirenz has quite a long half-life, and this leads to a monotherapy period where the virus can easily start replicating under the pressure of only 1 drug, and it only takes 1 mutation to develop resistance to efavirenz. A classical one is, for example, K103N. This resistance mutation is also very readily transmitted because it does not confer a very significant fitness cost to the virus. So we often see people with isolated K103N that's been transmitted. This contrasts with a patient who has taken antiretroviral therapy first line and then just stopped for whatever reason. For example, they moved house, they had an interruption in lifestyle, and these people often represent to the same clinics or different clinics without disclosing that they've already had first line, and they get treated with exactly the same drugs. But of course, if they've taken that single pill of a tripler, they will have potentially high-level drug resistance to at least 1 drug. But of course, we won't know that because we don't do drug resistance testing at baseline, and they will get standard ART. And if you see next, what happened, the result of that is, of course, is that you can see here that drug resistance in people presenting for first-line care, in other words, pre-treated, or these people with transmitted resistance, you can see here that over time on the x-axis, the prevalence of drug resistance on the y-axis has been increasing and is now sort of an exponential phase in sub-Saharan Africa in particular. And this is probably attributable to the rapid scale-up of ART, which has obviously saved many millions of lives. The downside is that, of course, you have applied large population-level pressure of drugs with a relatively low barrier to resistance because of the pharmacokinetics I showed you. So we have a problem of increasing drug resistance. Well, why is that a problem? Well, these data here show you that if you have pre-treatment drug resistance, you can see that the odds ratio of failing your first-line regimen are somewhere between 2 and 2.5. So you have between a 2 and 3-fold risk of virologic failure in your first year if you have a drug-resistant variant at baseline. So this does make a difference on outcomes, certainly in the shorter term. The right-hand figure shows you some similar data from a European cohort from back in 2011. So whether you're in Africa or whether you're in Europe, it's a similar story, that PDR increases virologic failure. What about mortality? Well, we looked at this recently in a study done in hospitalized patients in Malawi, where the patients had gone into hospital for various different reasons: TB, pneumonia, cryptococcal meningitis, and many other conditions. And we found that those individuals with a drug-resistant virus that was resistant to at least 2 drugs conferred or was associated with an increased mortality in a 30-day window period after their admission. So if they went home, they were 1.5 to twice as likely to die if they had drug-resistant viruses. And this is interesting because we used to think that just having uncontrolled viremia was the main risk factor. And we associate uncontrolled viremia with people with no resistance mutations because they've taken very little drug. But of course, these are people who have obviously been partially adherent to their medication, have developed resistance mutations, and still seem to have an increased risk of death as compared to those who had wild-type virus. Of course, these patients were on treatment for a little bit longer than the people without resistant virus, but nonetheless, these data suggest that we can use drug resistance monitoring or surveillance with point-of-care tests that are in development is a way to identify high-risk individuals in whom interventions can be applied, for example, adherence counselling or alternative regimen approaches. Next slide, please. So WHO did an analysis of what NNRTI drug resistance would do if nothing was done about it. And here you can see that nearly 500,000 deaths would have been attributable between 2017, when the analysis was done, and 2021, and potentially 300,000 new infections. So these were quite stark data about the rise of NNRTI resistance. And therefore, the question started to be asked as to what we would do about it. Next slide. And what WHO decided to do is to change the first line from NNRTI-based drugs to integrase inhibitor-based regimens. This was done with good clinical trial evidence from Europe, but there were actually no data from sub-Saharan Africa that this would be an effective option in that scenario. So it was a fair risk that the WHO took to recommend integrase inhibitors, in particular dolutegravir. So how do integrase strand transfer inhibitors perform in Africa? Well, there were 2 big studies. I'm going to just show you one of them. This is called the ADVANC study, done in South Africa, a large study published in the New England Journal last year. And you can see here in the left-hand figure that the standard of care in green, which was a efavirenz-based therapy, achieved an 80% suppression rate in intention to treat analysis, whereas the dolutegravir-based regimens, there were 2 of them using either TDF or TAF in the backbone, had somewhat slightly better outcomes. But if you looked on the people who were actually retained in care and taking their medication, there was no difference in efficacy, as you can see on the right-hand side. So we think that this is most likely due to patients with efavirenz side effects dropping out of the study. or not staying in the study. But of course, there's a problem with dolutegravir in that if you look at this graph, the weight gain in dolutegravir treated patients was fairly significant in terms of in comparison to the standard of care group. Now, of course, weight gain in a patient with HIV and wasting syndrome is a positive sign, but most of these individuals were not underweight at trial entry, and these patients, remember, were treated at fairly high CD4 counts. So very few, in other words, very few of them had CD4 counts below 200. So this is a signal that has been replicated in other studies, for example, the NAMCEL study in West Africa, that dolutegravir is associated with significant weight gain that now extends out to 96 weeks, and it can be considerable. So we need to weigh up the cardiovascular and renal consequences of this against the perceived benefits of dolutegravir. So we have undertaken some work to understand whether outcomes of dolutegravir will be compromised by drug resistance. Now, you would imagine NRTI resistance to affect the backbone, because we're treating not only with dolutegravir but also with tenofovir and a cytosine analog. But we also were looking at whether NNRTI resistance could have an impact on the outcome of integrase inhibitor-based therapy. On the left, you can see the proportion of patients in the two arms of the study of ADVANC and the prevalence of drug resistance, and it was between 10 and 15%, as you can see. The astute amongst you will notice that there was a higher prevalence of drug resistance in the dolutegravir arm compared to the efavirenz arm, and we don't know why this is. A number of ideas have been bounded about, but remember, this was a randomized controlled trial, and both arms should have had exactly the same prevalence of drug resistance at baseline. But nonetheless, on the right-hand side, you can see that if you had NNRTI resistance, in other words, the purple bars, you had a lower likelihood of achieving virological success. And the reason there are 4 panels there is that each panel shows the definition of virological success in a different way. There are different ways that clinical trials do this, but whichever definition of virologic success you use, the patients with drug resistance did significantly worse. This is interesting because, of course, none of these patients had integrase resistance at baseline. Although we did not formally sequence them, we know that the prevalence in South Africa is less than 1%. So there's another explanation here. And in the next slide, you'll see some of the possibilities. Did we not completely adjust for adherence, and is there residual confounding? The analysis I've showed you were adjusted for not only pill counts, but also subjective adherence measures. And so we think that the effect of residual confounding is unlikely to be big enough to account for this significant difference we found. Alternatively, one might postulate that drug-resistant virus has gained increased fitness, and we will need to investigate this using cell-based models and sophisticated models of replication. And these differences in fitness may not be immediately obvious in our typical typical assays that are very simplified in the lab. Of course, there's the potential for an unrecognized relationship between NNRTI and integrase activity. Of course, if you inhibit RT, you, by definition, affect integrase or integration. So, of course, there's a direct relationship between the two, but it may be a bit more complicated than that. But whatever the explanation for the findings, what we have come to conclude in that study, which is still under review, is that having drug resistance can be used as a marker beyond viral load, which can actually stratify patients into those who you may be more worried about. And we have a number of technologies to do this at point of care. They're not in commercial development yet, but they certainly could be. And so therefore, you might imagine a scenario where we do a point of care assay on each person starting ART and to identify if they have certain key mutations. And if they do, one may institute increased adherence or more frequent follow-up visits for those individuals to prevent them experiencing virologic failure. And this is a way we can almost tailor-make ART in order to ensure that we achieve the UNAIDS target goals. Just finish on a bit about long-acting agents. There's a lot of excitement in the field about long-acting ART. The integrase inhibitor cabotegravir is very similar to dolutegravir and has been tested in a number of studies in the last 5 years. The cabotegravir is co-formulated with a second-generation non-nucleoside called ripivirine. You give it by injection, but it's a fairly large volume, 1 ml for each drug. So that could have problems in terms of long-term adherence. But the latest data suggests that if you have an injection of these two drugs every 2 months, you have outcomes that are equivalent to taking triple ART, so standard of care. So this is an excellent result. It'll remain to be seen as to what happens in terms of the scale-up. Just a word of caution, of course, here on the next slide is that some early data from these studies on injectables showed that in Russia, three of the virologic failures who had a polymorphism in integrase at position 74, so L74I, these individuals experienced virologic failure with the addition of major integrase mutations. Now, it's unclear as to what the risk ratio was for having L74I and drug resistance and failure, but it is quite striking that each of the three virologic failures developed a major integrase mutation. We often see virologic failures without major integrase mutations, but in this study, they've shown these patients have clearly developed major drug resistance, conferring decent levels of resistance in vitro. So next slide, please. So this is something to watch out for. We did an analysis in Nigerian isolates, which are subtype AG or G, and we indeed found significant levels of this polymorphic residue isoleucine at position 74 in those West African isolates. And therefore, we need to bear in mind that the barrier to drug resistance or susceptibility levels of these viruses may differ from the subtype B viruses that most of the in vitro work on dolutegravir had been done on previously. Other long-acting agents in the future include one from Gilead, which is a very exciting inhibitor of the capsid assembly process. This has amazing pharmacokinetic properties by which you may imagine weekly dosing of tablets, and this could be combined with drugs such as EFDA, which is a Merck drug that impacts reverse transcriptase translocation, and this can also potentially be dosed weekly. But of course, these two drugs are very exciting on their own, but we need to see how the combinations fare in clinical trials. So in summary, I think I've hopefully convinced you that surveillance for drug resistance is important, because if those studies had not been done, then we wouldn't know why treatment failure was occurring and what to do about it. We know that integrase inhibitors are highly potent. They are the new standard of care, although they do have some drawbacks. One of them may be the fact that pre-existing polymorphisms in different subtypes may confer reduced susceptibility or facilitate emergence of major drug resistance mutations to integrase inhibitors, and we need to be very careful and watch for these mutations to emerge. And of course, this also demonstrates that ongoing work on drug resistance is vital, even though we have incredibly potent drugs. And just like to take any questions now. Thank you for listening.